Hello and welcome back to biology. So we talked about glycolysis and kind of the introduction to cellular respiration the other day. Today we're going to dive into kind of the aerobic side of respiration with the Krebs cycle and then the electron transport chain. So let's just dive right into it. So we got the Krebs cycle is the next step after glycolysis. So after glycolysis, we have pyruvate that's made from glycolysis. That pyruvate is going to enter into the matrix of the mitochondria, where it's going to go through the Krebs cycle. Krebs cycle is named after Hans Krebs. He was a German scientist who discovered the cycle. He actually won the Nobel Prize for it in 1953. He won the Nobel Prize for this. As we talk about the Krebs cycle, it's not necessary to know the individual steps of this cycle. All right, so as long as you get the big idea of what goes into it versus what comes out of it, you'll be all right. So pyruvate enters into the mitochondria, enters the matrix. What happens is we're gonna oxidize glucose. So we're gonna finish oxidizing glucose, which just means we're gonna move electrons away from it. We're going to finish oxidizing glucose, which is in the form of pyruvate now, and we also get carbon dioxide from this. So the carbon dioxide that's made from cellular respiration is made during the Krebs cycle. So pyruvate, before it actually enters the Krebs cycle, it has to go through kind of a pre-Krebs little thing here. What happens is that the pyruvate here actually has to change into acetyl-CoA. Part of what that change happens is pyruvate's a 3-carbon molecule, so acetyl-CoA is actually a 2-carbon molecule. So part of that going from pyruvate to acetyl-CoA is losing a carbon. Well, that carbon's going to get picked up by oxygen, making CO2. We also form another NADH during this process. So simply put, pyruvate's converted to acetyl-CoA. In that production, we say we get 2 CO2 from right here, 2 NADHs, and 2 acetyl-CoAs. Now, if you're like, well, hang on a second, I just see one here. Pyruvate's moving to acetyl-CoA. I see 1 acetyl-CoA. I see 1 carbon dioxide, and I see 1 NADH. So where are the 2's coming from? Remember that when we broke glucose apart in glycolysis, we've actually made 2 pyruvates. So there's 2 of these pyruvates. In each pyruvate, 1 pyruvate makes 1 of these. But because we have 2, we say that per glucose molecule, per the whole glucose, we make 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoAs. Once we have acetyl-CoA, then it enters into the Krebs cycle. You don't have to, know the Krebs cycle. You don't have to know all these steps here. But you may also hear it referred to as the citric acid cycle. That's because the first kind of molecule that's made in the cycle is citric acid. So this carbon compound is actually citric acid. So then we run through the Krebs cycle. The whole point of the Krebs cycle is to make this NAD, this NAD, and this fat. Now, we discussed yesterday how NAD is an electron carrier. It's going to carry electrons, right? So we have to bank electrons so we can use them during the electron transport chain. FAD is going to do the same thing. FAD plays the same role in this as what NAD does. So FAD and NAD are just the same molecule, or different molecules doing the same purpose. So if we look, we say that per glucose molecule, the Krebs cycle, we get 2 ATP, 6 carbon dioxides, 8 NADs get made here, and then 2 FADs. But if we look, let's just look at the carbon dioxide. So we say 6 carbon dioxide gets made, but if we actually look, we get CO2. 1 CO2 forms there, 1 CO2 forms there, and another carbon dioxide forms there. There's only 3 carbon dioxides that form. So where do we get 6 from? And in fact, if you look at all of these numbers, we only see half of them actually here. 1 ATP, 4 NADs, and only 1 FAD. So why are all these numbers doubled? Remember, we make 2 of these. Because since we had 2 pyruvate, we actually make 2 acetyl-CoAs. So that's why we say it's per glucose molecule. Because per glucose molecule, we get 2 pyruvates, which means we get 2 acetyl-CoA. 1 acetyl-CoA will make, 3 carbon dioxides, 4 NADs, 1 FAD, 3 carbon, 1 ATP. The other acetyl-CoA will make the same amount. So that's why we say it's per glucose, we get this many. So the whole point of that was for us to gain a lot of NAD and gain a lot of FAD. So what do we actually do with the NAD and the FAD? Well, then we move to the inner membrane and we go through the electron transport system, which is we call it oxidative phosphorylation. What that means is that by the NAD and the FAD giving up electrons to a bunch of these enzymes here, these electrons are going to move through these enzymes. So they're going to move through these enzymes. When they're moving through these enzymes, they're releasing protons out into the inner membrane space, which is the space between the outer membrane and the inner membrane of the mitochondria. So because of that, we have an electrochemical gradient now, because this side is positively charged, this side's negatively charged. Well, it doesn't like that, so they're going to try and rectify that. So these hydrogens are going to try to rush back into the matrix. So they're going to move through ATP synthase. Well, as they're moving through ATP synthase, they actually give energy to ATP synthase, to turn ADP into ATP. So then here's where we get this ATP part of it now. We made some during glycolysis, we made some during the Krebs cycle, but this is where we're going to make most of them. Now, if you're playing the home game, you're wondering, well, you talked about oxygen and water. Oxygen is a product, or as a reactant, water is a product. But where do they come into play at? Well, they come into play right here, because the final electron acceptor here is oxygen. Oxygen's the final electron acceptor. So as the electron's getting bounced around and bounced through all of these enzymes, the last thing that's going to accept it and take it is oxygen. Well, when oxygen accepts that electron, it actually breaks apart, because oxygen is always found as a buddy. There's always another oxygen molecule with it. Well, that electron's going to break apart those 2 oxygen molecules into single ones. Well, oxygen does not like that. It's not like being by itself. So because of that, it's going to bond, or it's going to pair up with the hydrogen that's entering via ATP synthase. When it does that, we get water. So the oxygen that we breathe in is used as the final electron acceptor. It's the final step in this puzzle here. And then that oxygen we breathe in is actually used to make the water that we see in this reaction. So how much ATP do we actually get out of this? We've been keeping track of numbers and all that stuff, but how much ATP do we actually get out of this? Well, let's figure this out real quick. So because we made ATP in a couple of different places. We also got to keep track of our NADs and our FADs, because the NAD and the FAD is what's going to give us the ATP at the end of this. So glycolysis, 2 ATP, 2 NADs. Krebs cycle, 2 ATP, 8 NADs, and 2 FADs. So at this point, just between glycolysis and Krebs, we get 4 ATP, 10 NADs, and 2 FADs, right? So now we gotta talk about, well, how, and then the NADs and the FADs move into the electron transport chain. But how much energy, or how much ATP does each NAD or FAD make? Well, NAD can make 2 or 3 ATP. FAD can make 2. So FAD is slightly worse than NAD. NAD's like the better one. FAD's is slightly worse, and that's because FAD enters the electron transport chain later. Because it enters the electron transport chain later than NAD does, it makes it slightly worse at making ATP. So that gives us 4 ATP, 10 NAD, 2 FADs, right? So theoretically, then how much is made during the electron transport chain? Well, let's use 3 for NAD. Okay, let's say for every 1 NAD molecule we get 3 ATP. Well, then we just multiply 3 by 10 NADs to give us 30, and the 2 FADs give us 4 ATP, so we get 34 ATP. When we combine this with the 4 ATP that's made from glycolysis and Krebs, we get a grand total of 38. Now that's the theoretical yield. Reality is there's not many things that actually make the full 38 ATP. A lot of times we're down 30, 32 when you factor in the ATP that must be spent in this process, because we had to spend energy to make this work still. So if you factor in that, and factor into that there's different bacteria that use different enzymes, so this number varies among different organisms because depending on the enzymes that are used in the electron transport chain, it will change this number. The other thing is oftentimes the ATP that's made is not a whole number. All right, sometimes we can get half an ATP or whatnot. And for this, we use 3. We're saying that each NAD makes 3 ATPs. Well, what if NAD only makes 2 low? Well, then this changes from 3 to 2, which changes this to 20. Oh, my mouse is not working right now. Changes that to 20. So now instead of having 34, I have 24, which changes this to 28. So it's very rarely do we get 38 ATP. So that's it for this video. Let me know if you have any questions, and I look forward to seeing you next time. Hope this was helpful.